this is a series where inviting faculty members and PIs at early stage of their careers to share both their past works and the vision they're pushing the field towards. And this is not just about being early career. We believe we are bringing people who keep delivering innovative ideas, who have been inspiring us with new insights and high quality works year after year, especially in this recent fast moving wave of protein design and molecular machine learning. And another big motivation you will see in the later schedule, many of our speakers are rising star PhD students and postdocs who are going to on the job market soon, and they will try practice their job talks here. And we hope this platform can give them more exposure and opens up new doors for their next move. So we have been planning this series for a long time, and we hope you find it useful and exciting. With this, let's save the time for science. I'll pass it to Divya to introduce our first speaker today.
Hi everyone. Thank you, Leo, for introducing this series, and we're super excited to introduce Professor Mohammed AlQuraishi. Dr. Mohammed AlQuraishi is an assistant professor in the Department of Systems Biology and a member of Columbia's program for Mathematical Genomics, where he works at the intersection of machine learning, biophysics, and systems biology. He earned an MS in statistics and a PhD in genetics from Stanford University. He subsequently joined the Systems Biology Department at Harvard Medical School as a Departmental Fellow and a Fellow in Systems Pharmacology, where he developed the first end-to-end differentiable model for learning protein structure from data. Prior to starting his academic career, Dr. AlQuraishi spent 3 years founding two startups in the mobile computing space. He joined the Columbia faculty in 2020. Prof. AlQuraishi's recent work includes OpenFold, a widely used open source reproduction and extension of AlphaFold, and Genie, a diffusion-based generative model for protein design. A pioneer in the field of computational protein modeling and design, it's an honor to open our series with him. So, Professor AlQuraishi, you can get started whenever you're ready.
Excellent. Thank you so much for the lovely introduction, and really a pleasure to be here to start off the series, which looks excellent. The lineup looks amazing, and I look forward to attending future talks as well. So what I'll talk about today, I thought, is essentially a kind of a series of observations that's come out of some of the work in our lab, but also more broadly in the field as a whole. So I'll cite work from many other labs as well, on sort of two broad sets of questions. One is essentially how AlphaFold and AlphaFold-based systems make predictions when doing inference, when a prediction is being made about structure. That's one set of questions. And the other set is the learning process, is how these systems essentially, as they're ingesting data during training, acquire knowledge about the objects that they're trying to model and predict, and how this sort of progresses throughout the process of training. So there'll be sort of two sets of observations that I hope get at that question in a way which I think is on sort of a lot of people's mind, as to the level of understanding, sort of physical understanding, other types of understanding that sort of machine learning models currently have of sort of these physical objects, right, of proteins and other types of modalities. So I hope this will be somewhat interesting, and yeah, let's get into it. Before I actually sort of really get into it, let me just kind of mention one broad thing, which is that my lab works broadly on these types of problems, right? So molecular modeling of protein structure, protein interactions, including other types of molecules like drugs, small molecules, nucleic acids, and so on. So we're broadly interested in all these sort of questions, even though the focus today will probably primarily be on the protein structure prediction sort of aspect of this. Although I do think now, as we are moving in this field toward other types of modalities, like small molecules, like nucleic acids, some of these questions I think will have slightly different answers, and I think that's going to be really interesting to sort of unpack over the next few months and years. Right. So let me kind of start with just a brief kind of review if people are not familiar with the space, that protein structure prediction has really undergone this revolution. That one way of sort of marking this progress of the revolution has been the CASP competitions, which happen every 2 years, that essentially assess the quality of predicted structures coming out of from models in general, machine learning or physics-based models. And just to say that sort of historically, this field has had limited success in being able to do protein structure prediction. But that really changed in the sort of the end of the last decade, where with the introduction of sort of the first AlphaFold and then subsequently AlphaFold 2, we saw this sort of very rapid progress in the ability of machine learning based methods to predict protein structure, right? And this is sort of measured here by essentially the RMSD, the root mean squared deviation of the quality of these predictions as they come out of those models. And this was sort of focused on proteins in this case. Now with kind of like the systems, we've gone beyond proteins to other types of biomolecules. I think the success that we're seeing with the newer models doesn't quite mirror what we've seen with protein structure, and maybe I'll get into that a little bit into why there is that bit of discrepancy. But I think suffice it to say that we are seeing kind of very rapid progress in the space, right? And in general, at least in the protein context, the success has really translated into predictions that are essentially almost indistinguishable from experimental structures, right? So these are very, very high quality predictions that really, I would almost argue, kind of push the envelope of what we would have absolutely I would have expected to be possible from any kind of data-driven approach. It almost kind of defies comprehension that one can get predictions that are this accurate. It still blows my mind, despite this having now been a few years old. So as I said about alluded to in the beginning, I'm going to kind of focus on two sets of questions, right? One is essentially and I'll focus on AlphaFold 2 for the most part, although when it makes sense, I'll try to kind of mention how AlphaFold 3 which is the latest system, might somewhat change these conclusions. But the first set of questions, like I said, it will focus really on, or the first set of observations, will focus on what AlphaFold like methods have learned, in particularly when it comes to this question of sort of physical understanding of the objects that they're trying to model. And then the second set of questions will be about the training process, how not what it has learned, but how it learns as training progresses. And so I'll walk through sort of these kind of five mini vignettes, if you may, that try to get at these various questions, starting with the question of, to the degree to which physical understanding has been learned by AlphaFold 2 and related methods. And this will actually center on sort of three observations that I think kind of collectively indicate, in my opinion, some measure of physical understanding by AlphaFold 2. So let's get started with it. Now, in case you're not familiar with AlphaFold 2, let me just give a high-level overview. 3 changes some of these things, but I don't think in a kind of fundamental way, excuse me. I would say the changes are fairly minor, so I think everything I'm going to talk about here more or less holds for the AlphaFold 3 context, but I'll point out when it doesn't. So you start out with these systems, right, with an input sequence, typically a protein sequence in this case. You're doing some sort of search against a sequence database that provides a multiple sequence alignment, so that's the top left, as well as typically a search against a structural database, most often the PDB, for related protein structures, right? Templates that can serve as essentially structural hypotheses as to the target that you're trying to predict. And then this process, proceeds through sort of 2 neural representations, the multiple sequence alignment and the pair representation, which is sort of an abstract representation of the underlying geometry of the protein. And this progresses through these sort of 48 blocks of what's called the pair former or the Eva former, which is essentially, kind of a neural network, some sort of transformer-based architecture that reasons on these 2 representations before it spits out the final structure at the end. One quick note here is that AlphaFold has a sort of additional bit where it sort of recycles those representations. So you can actually take the output, feed it back into the model, and sort of repeat the whole process multiple times. This does sort of induce a kind of an inductive prior, which is that for AlphaFold to essentially always refine structures, or to expect that the model is being presented with structures of varying degree of completion or maturity, and the model is acting to refine them. And the reason I think this is the case is because since you're recycling the output of one sort of run or inference pass through the model back into the input, you are at some points in time challenging the model essentially with seeing both very sort of nascent structures that haven't yet been refined to sort of high degree of accuracy, as well as highly refined structures, right? So the model, even at the very first layer of this sort of Eva formal block, is being presented with this sort of varying kind of degree of structural refinement. And because of that, the model, I suspect, is always being induced to essentially act in this kind of refinement way of being able to take any structure and then just make it a bit better. Right, a bit like I said, a diffusion type model. Even though AlphaFold 2 at least didn't really have any explicit diffusion. And even with 3, the diffusion piece is only in the structure module and not the whole structure. But anyway, that's sort of the high level overview. Now let me get into the question I started with, which is the measure of sort of physical understanding that the model has acquired. So the first observation that I sort of want to talk about was actually done by Sergei Ovchinnikov's lab at MIT now, where they essentially started out with this sort of basic hypothesis, and that hypothesis was um, you have a multiple sequence alignment. That multiple sequence alignment essentially kind of sculpts the overall energy landscape of the protein of interest. So sort of it provides a kind of a global overview of that landscape to the model, right? And this is a hard problem because you have a very large search space, and so being able to sculpt that overall landscape is key. And then that's sort of coupled now with essentially implicit structural or physical understanding by the model, some sort of scoring function that takes this global picture, right? And you can sort of imagine that as a sort of coarse positioning of the structure in this overall landscape of the conformation in this overall landscape down to the lowest energy state, right? So you go from what is, in effect, a sort of a coarse, low resolution structure through the implicit scoring function to a high resolution, precise atom level representation of the structure that's actually finally predicted by the model, right? So this is sort of the hypothesis that the MSA provides this kind of high level view, and then there's an implicit scoring function that provides the kind of the high resolution refinement of the structure into the final prediction. So at this level, it's just a hypothesis, but I think what they did was really clever. And this remains, I often say this remains sort of my favorite work in the kind of the post-AlphaFold world, and inclusive actually of the AlphaFold 3 paper. I think this really is just incredibly kind of inquisitive work done by the Ovchinnikov lab. So what they did was take the AlphaFold architecture, and then essentially ablate the MSA component. So remove the MSA from the picture. And the reason why this is critical to understanding the degree to which this notion of implicit physical understanding is present is that the MSA, per their hypothesis, is essentially providing the model with this very strong prior as to the overall global landscape. And you actually see this in the way AlphaFold works when you make predictions with it, is that if you provide with an MSA, and then let's say at the same time provide it with a structural template, right, here on the bottom, that is highly contradictory to the MSA, the model simply ignores the structural template. So the MSA is just a very dominant signal to AlphaFold when it's present. So by removing the MSA, you essentially remove this kind of strong prior, and the model is left to its own devices in the sense that it has to rely on a implicit scoring function, right, to be able to sort of judge the relative merit of different conformations given a protein sequence, right? So the way they set up this experiment is you still provide a protein sequence to the model. Obviously, you provide a structural template. You don't provide the MSA, so single sequence. You provide a structural template that essentially acts as sort of a hypothesis to the model as to what the prediction ought to look like. They run it through the inference procedure, and then they quantify the degree to which the model is essentially accepting of that hypothesis for this protein. And the way they do this is essentially based on the confidence score that the model assigns to its own prediction. Yeah, so AlphaFold is calibrated for every prediction, gives you kind of a confidence score. And so what they do in this case is to say, all right, if I provide a template that is of some measure of homology to the actual structure that I'm trying to predict, and then observe the confidence that AlphaFold has after having made the prediction, based on this template, that gives me some measure of the physical understanding of the model. So let me kind of make this a bit more concrete. So the experiment is basically as follows. So each dot here is basically a prediction coming out of AlphaFold. And in particular, this prediction is sort of predicated on a template, a structure that's given to the model. Now those structures that are given to the model, so each dot represents a different experiment, but for the same protein target. So the protein sequence is fixed, the actual sort of ground truth structure is fixed, but we are considering different sort of structural hypotheses, right? And these hypotheses can range from being very similar, that sort of on the right, where what's called the DKTM score is high. So this is, these are templates that are highly sort of similar to what's being predicted, to the left, where you have templates that are very poor, right? They're not good homologs, they're not good hypotheses to the structure that you're trying to predict. So the x-axis sort of spans a broad range of hypotheses from good to bad, right? And on the y-axis is essentially a ranking of AlphaFold's sort of happiness for that given hypothesis, right? So what this is saying, guys, given the high correlation, is that when you provide a good hypothesis, AlphaFold seems to be pretty happy, and then when you provide a bad hypothesis, AlphaFold seems to be quite unhappy, right? And again, this is absent the MSA, so the model does not have any access to the kind of the underlying coevolutionary structure of the protein, and so it is here having to rely on a simplistic scoring function, right? And the fact that the decoy TM score is highly correlated with the AlphaFold's confidence suggests that the model does understand, some notion of what makes a good conformation for a given protein sequence, right? So it understands that for certain sequences you want this conformation, and for other sequences you want that other conformation, right? So that sort of correlation is in their argument, and I buy it, is an indication of AlphaFold's sort of degree of physical understanding. So that's one observation. The second observation actually came out of from the initial paper that DeepMind published on AlphaFold 2. So again, coming back to this architecture, just to remind you, right, is that I said there's this evoformal or pairformal block which does the bulk of the neural reasoning in the system, right? And just to kind of recall is that first you have sort of 48 of these blocks. But in fact, they're repeated 4 times in the kind of standard AlphaFold 2 workflow. And so you have altogether 192 essentially layers through which a given structure is being processed, right? So what's interesting now is the following. Now, and again, this was actually in the original paper. So you could essentially put what they call a structural probe, which is a sort of a mini network that tries to predict from each of these layers the kind of the nascent structure, the structure as the model has understood it at that point in the neural network layer, or in the neural network the whole neural network. And I went backward. So what you can then do is essentially produce those structures, those predictions, as a function of the layer of the network, and then visualize, right, what those predictions look like as you proceed through the layers of the system. What I'm showing here now is a prediction, one such prediction, at that very first layer, right? So this is essentially the very first pass, and then the very first layer of that pass for this particular prediction target. Now, for this target, it just so happens to be the case that there's a deep multiple sequence alignment. So the model has a rather sort of strong prior as to what the underlying energy landscape looks like for this particular protein family, right? And that is consistent with the fact, right, that even from this very first iteration and the very first layer of that iterations, the structure already looks pretty good, right? It looks quite globular, and it looks to be pretty well defined. Now, if I were to run the animation of the predictions as you continue to progress through the multiple iterations and multiple layers of the network, what you see is that essentially the prediction is highly stable, right? That the model is not really making dramatic changes to the architecture. It looks a lot like that very initial prediction that the animation sort of started with at the very beginning, right? So what this indicates to some degree, right, is that when the MSA is deep, it looks to be the case that the model actually does not need all these neural network layers, that it sort of is able to, from the very beginning, essentially predict what that structure, at least roughly, ought to look like. So that's a case where you have a prediction target with a deep MSA. Now let's look at this other case where you have a prediction target with a shallow MSA. This actually came out, this was a Cas14 target, and this, I believe, was at the time from ORF8, which was a SARS-CoV-2 protein. And this was during the height of the pandemic, and at the time there was not that much sequencing data so for SARS-CoV-2. And so when they made this prediction, this was the DeepMind prediction, when they made this prediction, I believe they only had like 8 or 10 sequences in the multiple sequence alignment. So this was working with very limited data, right? And you can sort of tell that at this early stage, again, so this is the same story. First iteration, first layer of the network, so really very nascent structure. It looks quite loopy, right? It doesn't look particularly well. It doesn't look very globular. Now, if you were to now do the same exercise where you are observing the predictions from the structure as the model is progressing, you see profoundly more conformational sort of variability by the model, right? So it almost looks like the model is sort of considering alternate conformations and trying to sort of optimize some notion, right, of what is a good fold. To be clear, this is not, I don't mean this in the sense that this is sort of like an MD simulation where the model is actually minimizing some sort of physical or simulating some sort of physical trajectory. It's just to say that the process, right, appears to sort of be somewhat akin to a sort of a data-driven sampling procedure where you have some notion of physicality, and then the model sort of considering different conformations and then scoring them as the process continues, right? Which is very much dissimilar from the previous situation when the MSA was deep and was able to sort of rely on that covariation signal to predict the structure. Right. So that's another indication that the model may have learned some measure of physical understanding. Now let me sort of end with the third observation, which is something more of a minor one, a bit more of a technical story, but it's still quite interesting. And this I think came out during the Cas15 results actually, where there's a focus on on the prediction of multimeric complexes, guys. So now we're not predicting individual protein structures, but dimers. And in particular, in this process, there's a sort of a slight subtlety, right? Because you start with two sequences. You do again a sort of a search against a sequence database. You get two MSAs, one for sequence A, one for sequence B. But because you ideally want to identify the covariation signal between those two proteins, the standard procedure then is to not simply feed these two MSAs as sort of independent objects to the model, but to pair them across species, right? So now typically what's done is that the model is provided with a sort of paired MSA in which sequence A and sequence B come from the same organisms or in the reciprocal best hit within each organism for the query sequence, right? And this is what's fed into the model to predict the complex. So this sort of makes sense, right? And this is this is not dissimilar from a numeric prediction where again you're relying on this covariation signal to make a prediction. What is, however, different in the experiments that were done at Cas15, particularly by Yang Zhang's group, is that they made many predictions for complexes. Let me just see if I have the slide. Oops, sorry. Right. There were many predictions made that in fact far exceeded the quality of the AlphaFold 2 Multimer prediction, which uses kind of paired approach. So the base circles here are the predictions. Sorry, just to clarify, each column here is a prediction target at Cas15. The y-axis is the quality of the prediction, 0 being bad, 1 being good. Each base circle is a prediction coming from the vanilla AlphaFold 2 Multimer pipeline, which does this kind of pairing of the MSA. And then the blue triangles are the best predictions that came out of Cas15, which you see are considerably better than the vanilla approach. And what was interesting is that one of the best groups, I think the second best maybe, or third, certainly one of the top three, actually did not do this pairing approach. They simply took the two MSAs independently and fed them into the model, right? So the reason I bring this up now, back to the question of physical understanding, when you no longer pair the MSAs, right? What this means is that while you will have information about covariation within a protein, so the interprotein information is still present, the interprotein information is no longer present, right? Because that covariation signal is for the most part going to be broken because you're no longer lining up the MSAs. Nonetheless, so in this sort of regime, the model has to rely on again its implicit sort of physical understanding to be able to put those dimers together. And the point of this particular exercise is that even when you do that, the performance, of course, they did other things here, just to be clear. So, I'm not actually saying, just to be clear, that there were gains by not doing this. That's not the case. The gains came from some other direction. But just to say that doing this approach does not actually hinder necessarily the performance of the model. The model appeared to be highly robust to breaking the kind of covariation signal across the pair of MSAs. So this is suggestive, again, of some measure of physical understanding that was gained by the model, right? And so independently, any of these observations may not be entirely conclusive, but I think sort of taken together, they do point in the direction of AlphaFold to having gained some measure of physical understanding, and that I think is quite interesting, right? To the degree in which one is interested in building models that don't just simply memorize the data, but really are able to generalize to sort of, well, I guess that's the topic of the next section, to sort of unseen regions of structure space, right? Now, to be clear, I don't think this. So all right, so in the next two sections, in 2 and 3, in 2, I'm going to show kind of evidence now of the ability of these models to in fact generalize to unseen regions of structure space. So that's sort of essentially positive or further indication of the first point that there's some understanding of physics potentially. But I do think the story is uniformly kind of positive. And the kind of the third point I'll come to, I'll show some evidence that this generalization does have limits. And in fact, there are certain cases, almost adversarial cases where the model really does fail. But let's continue with a kind of positive story for now. So the next question I want to get to, right, is slightly different than the previous one, which is, setting aside how the model is making its predictions, whether it's physics based or some sort of MSA based, the question is still pertinent as to the degree to which the model is able to generalize to unseen regions of structure space. Of course, I think it's fair to say that if we think the model has learned some physics, it ought to be able to do this. But of course, that might be contingent on the source of structures that it's seen doing its training set. And so there's still a question to ask in this regard. So at this point, I'll take a quick interlude and just to kind of mention OpenFold, which is sort of our effort at the time to produce AlphaFold 2. So when just to clarify here, one point of technicality, which is when DeepMind released the AlphaFold 2 system at the time, they released a code base where one is able to make inferences, but they didn't release a trainable the training infrastructure to train these models. And so our kind of first OpenFold effort was essentially on reproducing AlphaFold 2 and you can providing it as a platform for building new capabilities. This came out last year. Incidentally, we're sort of working now on reproducing AlphaFold 3 with OpenFold 3. I won't really get into it today. There has been several reproductions, I should say, or attempts at reproductions with AlphaFold 3. Although I do think there's still a lot to do, and we'll have more to say very soon on OpenFold 3. But for now, I just kind of want to mention briefly that what in this context of this conversation of this talk, what we used OpenFold for was to sort of probe this question of the ability of the AlphaFold 2 class of models to generalize to unseen regions of structure space. Just to say one other point here is that the way we sort of built this was through this kind of consortium that was sort of part academic and part industry, and it was sort of like collaboration that kind of bridges those two spaces with an intent of making this sort of have a steerable scientific focus so that industry has a say in kind of the kinds of problems that we pursue in exchange for supporting this sort of effort. And in particular, we've sort of been going after various problems that are of sort of interest to people who want to use these models in sort of production setting for certain industrial applications. Okay, but back to the main story. So just to say, with OpenFold that this was a completely new fresh code base, but we built it in a way that had essentially bitwise compatibility with AlphaFold 2. And what you see here is for each point is a prediction from AlphaFold 2 and OpenFold. The y and x axes are just the quality of the prediction. Again, 0 here is poor, 1 is good. Most predictions are in the top right corner, indicating high accuracy. But you see a sort of a spectrum of prediction qualities. The more critical point is just to say that they are highly correlated. So the two systems, essentially behave in the same way. On occasion, when they diverge, almost invariably, that divergence is just due to the inherent stochasticity of the prediction. So that there's not a meaningful way in which these two systems are sort of in expectation statistically distinguishable. And in general, for any given prediction, it looks a lot like this, where the white is the ground truth prediction, the blue is the AlphaFold prediction, and the pink is the OpenFold prediction. Okay, so now let me get into the main point. So what we wanted to do, right, is get at this question of structural generalization, and the way we thought to do it is the following. So at the CASP 14 competition, when kind of AlphaFold 2 debuted and when they made the big splash, the CASP organizers had gone and essentially curated data sets, right, protein targets that were very difficult. So they were difficult because they looked structurally different from what's in the protein data bank. Their sequences probably lacked, very deep homology. So they were the kind of targets that were expected to be difficult. And so that was an excellent experiment. It's I think demonstrated unequivocally that AlphaFold 2 was doing non-trivial things, right? That one could not explain its performance by memorization, right? Nonetheless, it did leave open the question of the degree of generalization. So another way of phrasing this is to say, if you sort of challenge the problem with increasingly difficult tasks where the training set looks increasingly dissimilar from your sort of test set, right? How well would the model do, right? If you gradually make the challenge more difficult. And this is what we tried to ascertain here. The first set of experiments was actually pretty straightforward. Here, we didn't even do any kind of structural stratification. We just asked, if you just had less data, so if you take the PDB and subsample it in smaller and smaller kind of portions, right? So the color coding here indicates a fraction of the PDB that was retained for training, pink corresponding to all of the PDB, and blue corresponding to the light blue, the bottom blue, corresponding to like less than a percentage point. So something like 1,500 structures at the time that were used for training this model. What would happen to the behavior of the model as you train it? And what I'm showing here are training curves. So the x-axis is the training iteration, the y-axis is the LDDT. Again, one is good, zero is bad. And to begin with, if you look at the pink curve, what you see is that the model is you see the behavior of the model essentially when the PDB is fully accessible. So this is like the default model, if you may. It achieves an LD to about 0.8 by the 7,000th step or so. It's worth pointing out that the model is trained for, I think, a couple 100,000 steps. So this is by no means, these are by no means fully trained models. These are all sort of early trained phases of the model. But given the expense, the compute expense involved in generating these curves, we couldn't actually train the model to completion every single time. So there is a caveat here, which is we are focusing on that kind of early training behavior. Nonetheless, what you see here is interesting, right? Which is even though the model, when you train it as 0.76% of the PDB, it drops in performance. Nonetheless, at the kind of the greenish, curve, this one, which corresponds, I believe, to something like 1 to 2% of the PDB, so maybe about 3,000 structures, the model is still reasonably performant. Right? The LDET that it achieves is about 0.7. It's not great, just to be clear. But it's actually better than the first AlphaFold. So the first AlphaFold that sort of debuted in 2018 and made a big splash, that one had an LDT of about 0.6, right? So already what this is indicating is that AlphaFold 2 is a sufficiently kind of more performant architecture, right? Just the design of the neural network, such that you're able to use 100 times less data than the first AlphaFold and still outperform it, right? So this is sort of quantifying the kind of the architecture gap. That AlphaFold 2 is about at least 100 times more data efficient because it's able to use 100 times less data and still outperform the first AlphaFold. So that's pretty exciting. But nonetheless, right, one could legitimately argue here, well, okay, you subsample the PDB by a factor of 100, but that doesn't really say anything about generalization. It just says you don't need a lot much data. But as long as your data is pretty diverse and pretty representative of sort of fold space, you can still make pretty good predictions, right? So the next set of experiments was to specifically address this, and that was to do the following: to say, okay, well, we have existing structural stratifications of the PDB, right? And particularly the one we use is called CATH, which sort of hierarchically structures the PDB at the class architecture and topology level. So it sort of starts at this kind of very high level, very coarse-grained stratification, which is based on alpha helices, beta sheets, and combinations thereof, all the way down to topologies, which are much more akin to like folds, right? And there are several hundreds of these topologies. So the question now was, well, what if we do the following? Let's start with topologies. What if we hold out certain topologies, or rather, actually, let me flip it. What if we train on a subset of topologies? And again, the same idea. So now when I say 10% or 20%, it's not 20% of the data in terms of like data points. It's 20% of topologies, right? So we're retaining, we're doing this accounting of the fraction that's used for training based on the classification, right? And then we test on some held out data set, right? That's topologically disjoint from this fraction, right? And so what you see here is a similar thing to the previous slide, right? Where sure enough, when you go all the way down to 5%, you do absorb a pretty serious hit. But already even at 10%, right, that's the green curve. This again is a model that is competitive. It's not only competitive, it actually outperforms AlphaFold 2, right? And even with 50%, right? So now again, this is you're sort of not seeing half of the fold space anymore, right? You are essentially almost indistinguishable from the full model, right? At least at this stage of training. So that's really encouraging. It suggests the model is able to generalize at the level of topology at least. Now, what's remarkable is that this seems to be true at the level of architecture. So even when you go kind of bigger groups, and I think there's maybe on the order of like 30 architectures in the PDB. So this is a really much coarser stratification of structure space. And then most surprisingly, even at the level of class, right? So here we're essentially training on just alpha helical proteins and testing on beta sheet proteins or the converse. And we see a similar situation where the model is able to generalize at some level of accuracy. And just to kind of communicate that visually, here are some examples. The top row, and these are not cherry picked. So I mean, the LDTs, you can look at the numbers, they're sort of hovering around 0.7, which is kind of the mean accuracy that was achieved by the model. Right? So these are representative examples. So what you see on the top row in orange is the prediction target. This is the ground truth. That's a helical protein in orange. And then the magenta is a prediction made by a model trained on alpha helical proteins. No surprise there, it does quite well. In green is a model that is trained on beta sheet proteins and then trying to predict this helical protein. And you see the LDT drop, it's 0.5, so it's actually worse than the mean accuracy. But nonetheless, right, and so it hallucinates a couple of strands, but the model sort of is not completely wrong. There's a lot that's correct about the structure. And then the converse is in the bottom, where you have a beta sheet target in orange. The green is a prediction made by a model trained on beta sheets. Again, not surprising, does quite well. But the magenta model, or the magenta prediction, is made by a model trained on alpha helical proteins. And yet, so not surprising here that the sort of the atomic bonding network, right, is wrecked. So PyMol no longer sort of recognizes the beta strands, and so that is lost, to be clear, that kind of atomic resolution. But nonetheless, that's the alpha trace, right, the kind of backbone of the model of the structure, the prediction, is sort of roughly correct. And again, the LDT is 0.66, which is sort of reflecting that kind of mean accuracy. So the model hasn't lost, entirely its capability to at least give sort of reasonable starting points as to the nature of the protein. And I, my guess is that this has a lot to do with MSAs, and I'm happy to kind of expand on this if people are interested in the Q&A. But just this to me was already sort of, this was very compelling. Right? This was surprising and not what I would have expected coming into this. We thought that when we do this, that the model would just completely get destroyed, basically. And so it was really kind of a pleasant surprise to see that it's able to generalize as much as it seems able to. Okay, so that's sort of maybe the end of the good news on at least this section. Now let me kind of end with a bit of bad news, which is at least one regime where the model does really seem to struggle to generalize to sort of structurally disruptive mutations, right? So these are predictions here where essentially you have one change in the protein. Right? So one amino acid. These are kind of depicted below, where we know those mutations are highly structurally disruptive. But where the prediction of the mutant form by AlphaFold essentially perfectly overlaps with the wild type form. So it really seems to be unresponsive to these kinds of to these kinds of challenges. And there was this other study which used a kind of a prediction of the delta G of sort of the energy and so sort of a similar effect. Now, what I suspect is happening here, right, is that because of the way these models are trained, because of the fact that they provide with MSAs, they are basically being challenged in the case of mutant forms to ignore their training, right? Because their training is essentially teaching them that within an MSA, within a family, you should have the same structure. That essentially the MSA sort of tells you what's invariant about that protein family structurally, and you should ignore, in effect, right, the sequence variation that sort of disrupts or that essentially disrupts that sort of underlying hypothesis, right? But of course, that's precisely the kind of mutation that you have in these instances where they are disruptive to the kind of structural integrity of the protein, right? And so it's really the sort of prediction task that is in effect adversarial to the way that AlphaFold was trained. And so I think for that reason, not surprisingly, the model really struggles to be able to do well here, right? And I suspect AlphaFold 3 and sort of future generations of these models will get better at this, but it does remain to be sort of an outstanding problem. Like that we really are quite incapable currently to sort of capture the structural impact of mutations on structure, particularly when they sort of shift the equilibrium from one particular conformation to another. Right? So where it's not simply a matter of saying, okay, the protein is unfolded now, but rather saying the protein has a new conformation that simply sort of that simply looks different, right? But in some sort of meaningful way, in some sort of gradual way from the wild type conformation. Okay, So that's it for the sort of the piece about what has AlphaFold 2 learned, and in particular how does it make predictions, right? Now let me kind of shift gears a little bit, and this will be I think a shorter section. Let me shift gears a bit and talk about the learning process, right? Because I think there too there's some interesting observations that tell us a bit about how these models work, and maybe provide some insight as to the question of introducing physical priors into these models, which I think has been a kind of a point of contention recently, in particular because the bitter lesson, as it's been kind of applied to molecular modeling, I think has been, obviously not just more data, but in a way weaker inductive priors, right? That we sort of we in a way want less physics, less physics based, not we don't want the model to learn less physics, but we want to bake in less physics. We want the model to learn as much as possible. And I think this first result in particular will maybe provide some explanation as to why at least in one particular case, that might be a good idea. So what I'm going to show you next, so just recall for a moment, right? I showed you an animation or a set of animations earlier, which were coming from a fully trained model, from a fully trained AlphaFold, but from a partial execution of the inference pipeline, right? So what we did in the previous set of animations was to say, all right, we're going to partially flow through the model and then sort of interject at some midpoint, and then take a structure prediction from that midpoint and observe it, right? I'm going to do now something quite different. I'm going to take so the inference pipeline here, right, is going to be complete. So all the predictions that you're going to see now in these animations are coming from the full pipeline. So this is not an intermediate prediction. On the other hand, the models that are being used to make those predictions are going to be partially trained models. So I'm not taking a fully trained model like I did in the first case. Here, I'm taking models that are either extremely undertrained. Well, as you'll see, we'll essentially trace the training process. If you recall earlier, this sort of curve that showed, very unperformant model and then sort of very quick rise and then kind of a saturation. We're going to kind of span that whole spread and then look at the predictions as they are being generated from this sort of this sort of exercise, right? So this is the first one, and what you see here, right, is this kind of progression from what's essentially a single point into kind of a 1D line, a 2D surface, something of a flat surface, a 3D volume, right? And then finally, sort of sufficient atomic resolution or accuracy, rather, so that you begin to see the details of the secondary structure elements being recovered correctly by PyMol, right? And this is not cherry picked. So this is one example. I'll show you another example with kind of the same phenomenon, where again you see this kind of progression from a point to a 1D line to a 2D surface to a 3D structure, right? So, and just to clarify, right? So this is an important point in that this is happening not at the level of the data set. So this isn't simply saying, oh, the model is kind of learning some sort of lower dimensional projection of the data set. This is saying that the model is learning a kind of a per data point projection onto these sort of lower dimensional objects, right? As proceeding through training. So let me actually just make one quick point here, which is, I started the section by talking about the fact that the kind of the bitter lesson in biomolecular modeling has been we want weaker and weaker physical priors, right? So this animation is sort of one as an indication of maybe one reason why you'd want that, which is to say the optimization landscape, right, that AlphaFold takes progresses through this extremely non-physical sort of intermediate phase, right? The predictions that are being made in the 1D and 2D phases are obviously very non-physical, right? So imagine now if we had some sort of physical prior that says you should never make a prediction that looks like that. Whether that's a good thing or a bad thing, we can at least agree that that would be highly perturbing of the optimization landscape that AlphaFold 2 takes. Because at least in this case, right, AlphaFold 2 seems to sort of like to go through those kind of non-physical intermediates. And I think that's at least one indication as to why it may not be so simple or so always beneficial to inject physical priors, because those physical priors don't just provide additional information, they also reorganize your optimization landscape. And that potentially could be negative. I mean, of course, it could also be positive, but just to say that there is that kind of potential impact, right? But let's come back to this result for a moment. So, so what I'm showing you here now is kind of a tracking of the model's performance as it trains, right? And this is basically the kind of the region of like the animations that you just saw basically span that region. So GDTS is the sort of the overall model performance. And then green is so the recovery rate of alpha helices, and then orange is the recovery rate of beta sheets. And what you see here, right, is that just as so the animation kind of anticipated, GTS, which is essentially a measure of the overall structural integrity of the protein, actually rises before there's this gap, right? Before alpha helices and beta sheets rise. So it's mirroring what we saw earlier, where global structure is recovered before secondary structure is predicted well, right? And another way to kind of quantify what we're seeing in terms of this kind of 1D, 2D, 3D thing is the following. So we can kind of consider on the left two extreme hypotheses. One hypothesis, which is maybe the one I would have sort of expected, is to say, okay, the model is trying to learn at every step information about the 1D, 2D, and kind of 3D aspects of a protein, right? It's trying to maximally extract the lowest hanging fruit in every physical dimension. If that were the case, we can quantify this by asking, what is the delta between the prediction of the model as is, the full prediction, versus a sort of a lower dimensional projection of that prediction, either down to 2D or down all the way to 1D, right? And that's what you're seeing in the top left. So the blue curve is the prediction versus the ground truth structure. There's no delta. This is a standard curve as you may expected. The green is basically saying I'm going to take the prediction, I'm going to flatten it into 2D, and then compare it against the 3D structure. So I'm losing the 3D information the model has, but by kind of tossing it out and then just looking at the 2D pancake. And then the red is doing that even further with 1D. So if you expect all spatial dimensions to be learned simultaneously, you would expect kind of all the lines to sort of continue to drop at some respective rate, and then flatten out because ultimately the 1D structure will never fully compare with the 3D structure. So there's always going to be a gap between the red and the green and the green and the blue. That's one hypothesis. The second hypothesis is that maybe the model essentially learns the 1D information sort of first completely before it transitions to learn the 2D information and then the 3D information. And that's what you would see in this kind of perfect staggering, where essentially, right, in the first phase, there's no daylight between the 1D, 2D, and 3D projection because they all are just like 1D, right? And then once the 1D information is sort of fully extracted, you then move on to the 2D and then you move on to the 3D. And what's interesting, so these are sort of hypothetical cases. What you see empirically is right? Which, at least to sort of my eye, looks a lot more like the staggered approach than the no staggered approach, right? So this to me is sort of not trivial, that the fact that the model isn't simply trying to extract all the information that there is at every dimension, but instead is some kind of almost staggering, I guess, of the learning of the dimensions, right? And not to belabor the point, but as I said earlier, this clearly has some implications as to the nature of the optimization landscape and how the model sort of learns to actually behave, right? Now, what is interesting, I think, here is the degree to which this sort of approach is an artifact of the AlphaFold 2 architecture specifically, or whether it sort of holds true more generally. We had built models in the past for protein structure prediction that behave very differently than this. That they don't have this kind of this sort of intermediate stages. In fact, some of the models we had built, which actually had a stronger physical prior, tended to learn what I called sort of slinkies in the beginning. So these sort of kind of very spread out helical structures. These were kind of the early predictions, and then they begin to sort of mold them to look more globular. It's in a way a bit of an open question what AlphaFold 3 learns, and that's something we'll be doing in the context of OpenFold 3 to try to understand its learning behavior. But just to say that, at least empirically, looking at other models, we don't necessarily see this behavior all the time. Okay, I'll end with the last bit, and just kind of going a little bit back to this question of the MSA versus the implicit physics, but now coming to it from a new prism, which is the prism of like learning. How does the model learn as you progress? So one thing I sort of skipped over or didn't really talk about in the beginning was the overall behavior of the model. How does it actually acquire knowledge as it trains, right? And just the one quick point here is that the model has this sort of rapid rise phase where in the first 5,000 or so steps of training, it essentially acquires about 90% of its final accuracy. And then the remaining sort of 90% or 80% of training give you that last 10%, right? So you go from point A to 0.9 LDT or something like that, right? So there's this sort of rapid rise and then kind of slow learning, right? Um, so at a sort of at a naive level, this seems to suggest that, well, you actually don't need all this additional training, that you sort of get a pretty good model pretty early on. But I think I think that's true. But it doesn't necessarily differentiate between the kind of two modes of predictions that I sort of talked about earlier in the very beginning, the kind of the MSA-driven approach, which is relying on this covariation signal, versus the kind of the implicit scoring function approach, which is relying more on the model's understanding of the underlying sort of fold space and kind of the sequence structure patterns that underlie protein folds. So coming back to Sigav-Chennikov's experiment, right, into the kind of ranking ranking these models, what we did was basically kind of redo this experiment that they had done, but now using these partially trained models. So what we want to ask right now is how good is the model's implicit physical understanding as a function of training iteration? And what I'm going to show you next is basically a plot of the correlation that of this figure, so just quantifying the correlation as a function of training time. And so what you see here, which is quite interesting, is that even by the 10,000th iteration of model training, where here you see that the model is doing quite well. Right, this is maybe even like 0.85, so it's almost 95% of the way there in terms of model accuracy. The correlation with respect to this experiment of kind of quantifying the model's ability to rank different structural hypotheses is about 0.7. It's not terrible, but it's not anyway close to its final correlation, which is north of 0.9. I think actually some variants of these models could reach 0.95 correlation, right? So this is actually suggesting that this sort of long phase of training here, while may not necessarily improve kind of the model's overall accuracy under most circumstances, at least in regimes where you sort of are more reliant on this kind of implicit scoring function, this becomes much more important. And that's something which I think is quite interesting and wasn't sort of evident just from the model itself, obviously, because we didn't really understand how it behaves as you progress through training. So I think I'll stop here. I just mention the folks who actually did all the work, particularly Gustav, Christina, Sasha, and Nazim, who led all the work on OpenFold, as well as sort of funding agencies that supported this work and private organizations and companies, including several who sort of comprise the Open Molecular Structure Foundation. This is kind of support for OpenFold through the OpenFold consortium. And this is my lab maybe a year ago or so. So I'll stop here and I'm happy to take any questions.
Thank you so much. And yeah, if you guys have any questions, feel free to either unmute or drop it in the chat and I can read them out. Okay, we have one in the chat. Based on these observations you described, can you share any thoughts on the pros and cons of using AlphaFold with MSA versus in single sequence mode to screen designed proteins?
So designed proteins are sort of an interesting, unique kind of situation, which sort of almost kind of belies everything I said. In particular, so just to say one quick point. In general, when you're making predictions with AlphaFold with single sequences, right, you're typically getting actually very bad predictions. Just to be clear, so these experiments with the ranking and so on, they're artificial in the sense that you're providing the model with a good hypothesis. If you don't have a hypothesis and you just try to make a prediction with a single sequence, the answer you're going to get, generally speaking, is quite poor. Design proteins, however, sort of live in this kind of interesting space where partly I don't think this is actually true of like every conceivably designed protein, but I think it's true of the proteins that we've designed in humans. They typically are coming out of some sort of Rosetta energy, function optimization procedure, or more recently through protein MPNN. And what typically goes through that process, right, is sort of an optimization for sequences that are highly rigid and that kind of in a way maximally capture the sequence structure characteristics of a given fragment, right? So they're really sequences that are ideal in some way. And so in those cases, generally speaking, right, you're operating this kind of very strange regime where actually the single sequences are quite good, right? And so for that reason, design proteins, generally speaking, work quite well in this context. I mean, sorry, the predictions of design proteins using single sequences work quite well. we sort of you don't actually need an MSA, and it's sort of it's I think it was something of a surprise when these models were first being tested in this context that this was true. I think the broader challenge with design proteins in general, however, is you simply don't have access to an MSA. So that becomes the issue, right? So because if it's sort of genuinely kind of a de novo protein, then it's not going to look like a natural protein, and you're not going to have access to that sort of information. Having said that, I mean, I think if the process was like, let's say, something in which you're doing some sort of sequence optimization. So you have some sort of random library, and you're starting from a sequence and you're randomizing it, and you have some sort of assay that actually confirms that the protein is expressed and so on. When you are randomly varying those sequences, I think that sort of sequence collection is likely a good representative that you could use as sort of an artificial MSA for your protein of interest. So I think if that information is available, I would use it. But I think particularly in the protein design context, we are in this sort of funny regime where generally speaking, single sequences are actually okay.
Thank you. And we have another question. Is there any way to use AlphaFold for scoring without the need to push the structure as template?
So, I mean, generally speaking, right, if you give it no hypothesis, if you don't give any structure, you are going to get a prediction out. And that prediction does come with a kind of a calibrated self-confidence. So I suppose that is essentially a draw from this kind of sampling procedure that the model seems to be making. So that does work. The challenge, however, generally speaking, is that the space is too large, right? So kind of naive sampling is hard, right? You're unlikely to get a good prediction out. Um, there are some exceptions to this, right? So in the AlphaFold 2 context, people have seen, at least with maybe shallow MSAs anyway, that if you inject noise to like dropout at inference time, not just training time, you can sort of force the model to sample alternate conformations. You can kind of get it to behave differently every time you make a prediction. And by doing so, you could sample alternate conformations. Of course, you might need to sample a whole lot. And some of the results I mentioned earlier for the Cas15 competition with multimers, um, some groups, went to tens of thousands of predictions to be able to get good structures out. So there's that wrinkle. With AlphaFold 3, it does seem to work better in this regard. It seems to actually be able to better rank order its own predictions. And at least in the context in which this was tested, which is for antibodies, DeepMind did see increased performance as you increase the sampling frequency, all the way up to, in their paper, up to 1,000 samples. But the increase was sort of log linear in the sense that you go up sort of log uh, in number of samples, and you kind of gain linearly in the some sort of measure of accuracy, guys. So it's really expensive. It's an example of inference time scaling, obviously, for this problem, but it's like log linear, so it's not maybe quite the regime you want to be in. And I think one area which we're enthusiastic about is thinking about, can we improve the efficiency? Can we make these models be able to sample both more quickly, but also just more intelligently, so that you're not spending most of your time proposing kind of bad structures.
Another question asked if you're still pursuing research on mechanistic interpretability and how promising do you think this approach will be on these types of architectures?
Yes, absolutely. And I'm actually quite enthusiastic. So one reason why I'm enthusiastic about the mech interpretability for these problems is that we have good priors. We have actually good theories about how proteins fold, right? Physical theories. Very much unlike even in the biological context, like for instance, protein language models, and certainly in the natural language context, like with NLP or computer vision, where we don't really have good theories of the brain, or we don't have good theories of, like, say, protein sequence space. Protein structure, we actually do have good theories. We understand that protein folding is driven by hydrophobics, for instance, right? We understand that protein binding is driven by electrostatics, for instance, right? So a lot of these kinds of physical theories can be used as strong priors in terms of how to pursue and what to look for in the ability of these models to understand mechanistically what's happening, right? And so for that reason, I think if there's one particular domain where one can make progress in this space, will be either like protein structure or chemistry more broadly, where the underlying kind of scientific theories are well developed.
There is evidence showing that for ligands, AF3 is mainly memorizing. Could you comment on that as chemical space for ligands is much bigger than proteins, which are only composed of 20 amino acids?
Yeah, I think that's at least partially true. I mean, I think the experiments that have been done, and particularly maybe the best ones so far, have come out of Toshin Strait's group, the runs and poses experiments, where they do a bit like what we did, right? Where you sort of quantify the similarity of your test set to your training set, and as you take your test set further and further away from your training set, the performance drops. So in a way, I like what I showed, where the model was fairly robust. Here, the drop is much more precipitous. Which suggests, so I think saying it's only memorizing is too strong, but I think it's fair to say that it's some mixture of memorization and generalization, and it's nowhere near as robust as a protein case. Um, I think that the question already kind of alludes to the answer, right? Which is that the fact that chemical space is larger, and I think even more importantly, perhaps, that you don't have that MSA signal. So in a way, here you're sort of entirely reliant on physical understanding to make good predictions, right? The covariation signal is not present. It's not going to help you. So in the kind of the AlphaFold 2 case where I said, you have the MSA, you have the physical understanding, they kind of work together to sort of make you do better predictions or have better predictions. This is no longer true here, right? So here we're sort of purely leaning on the model's physical understanding and having to deal with a chemically much more expanded space, right? So we're sort of being affected in two ways, right? Which makes this hard. Now I will say, I mean, I think we have a couple of efforts that are trying to get at this. So, on the one hand, we have this sort of consortium that we stood up that is trying to train versions of OpenFold 3 on private pharma repositories. So where we have, presumably a much more expanded chemical space, and we can kind of leverage that data to train models, on private IP to be sure, but potentially at least demonstrate the utility of having larger data sets that go beyond what's available publicly. So that's, I think, one interesting direction. The other is I also think that there is increasing capability to generate crystallography data at high throughput, right? Particularly with Diamond in the UK, and so I do think we are going to be in a regime in the next year or two where the availability of data, even crystallographic data, probing protein and small molecule binding, is going to sort of improve by leaps and bounds. And that sort of opens up a new opportunity now to be much more thoughtful about how we collect data and how we train models that just capture a lot more of chemical space. So I'm not pessimistic. I think perhaps modeling alone can't get us there, but I think with sort of the new innovations on the experimental side, I do suspect we'll see continued progress over the next year or so.
Thank you so much. We have a couple more questions. How does single sequence mode perform on fusion proteins with two known proteins/domains? The problem was not well handled with MSA as it confused the resulting fusion structure with the known information.
Yeah, I think the problem. I mean, we haven't tried it, to be honest. So I don't have direct experience or direct, empirical sort of results to report here. But my guess would be is what would happen is that the individual domains will be reasonably well modeled potentially. But what's going to happen, I suspect, is that the relative orientations will not be, right? Because the model doesn't sort of have data on that. And I think that's going to be the challenge. If the scientific question is focused on sort of that aspect of it, the model might have some trouble. Now, to be clear, right? I mean, I forgot which, I think it was like the second or third section that I showed. The third set of observations in the first section that I showed had to do with predicting protein-protein interactions without sort of paired MSAs. So these models clearly have some capability to do this. So I don't think it's sort of hopeless, but I think it's a harder problem than would otherwise be the case for that reason.
Okay, and I think we have one last question, unless if people have others, feel free to drop them in the chat. But um, it seems like some groups have shown pairing was not that essential for Cas15 with AlphaFold 2. Has someone showed this for AlphaFold 3 as well? And what would that change about the conclusions and how about what this tells us about how the model is working?
So the closest to that experiment would be the antibody antigen results that DeepMind reported, right? In those cases, just by definition, you're having you're not having paired MSAs, and so they the model certainly still has predictive capability, better than AlphaFold 2's, certainly in that case, much better than AlphaFold 2. So it does seem to have actually done better in this regard. Although the precise experiment that the questioner is asking hasn't really been done in the sense that can you take just more broadly protein-protein complexes and then do this sort of experiment, which is an interesting one. One should do it. But my expectation will be that it actually does as well, maybe even better. And part of the reason why I say that is because in a way in AlphaFold 3, the kind of the center of gravity has shifted even further toward the implicit scoring function by virtue of the fact that they took the MSA module and shrunk it considerably, right? So in AlphaFold 2, the MSA module was sort of paired with the pair former, and he had 48 layers of MSA reasoning, while with AlphaFold 3, they just shrunk it down to like 4 layers, and they just did that almost like a pre-processing step. So for that reason, I actually think the kind of insofar as the MSA information is useful, which it is certainly, but it likely plays a smaller role in 3 than it did in 2. And so my prior would be that it will hold and it will be even more true. But I think that experiment is worth doing, and I haven't seen it done by anybody yet.
Awesome. If anyone has any other questions, feel free to unmute yourself. If not, thank you so much, Professor Al-Quraishi, for an amazing talk and for answering the questions. We really appreciate you being the first speaker in this new series. Set a very high bar. And thank you to all of you for joining. And thank you to Leo for getting this seminar series started and having the idea for it.
Thank you for having me. It was a pleasure. Take care.